Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
and Molly Kilty. Hello, everyone, and welcome back to another episode of our Jack Westin MCAT Podcast with your hosts, Mike and Molly. Mike, I feel like I always tell students what we talked about. I want to hear from you today. How are you? Happy holiday.
Oh my good. Oh, happy holidays, Molly. When this is coming out, I think it'll be the new year. I think this is going to be the first episode of the new year, which so happy new year, everybody. I hope everyone had a fantastic new year. Got to hopefully maybe get some rest, but also maybe, get a little bit of work done while we're kind of mixing in the rest. But we're kind of back on track in this new year, starting 2026 strong, really kind of piggybacking off of how we ended 2025. Because last episode we really focused on kind of how the body, especially mostly the female body, gets ready for pregnancy, right? Like how the uterine cycle and ovarian cycle coincide to form that overall menstrual cycle in order to kind of get ready, and we talked about what happens if pregnancy doesn't happen. The uterus is just so fed up. It's like, I worked so hard. I know if you're like, what are you talking about, Mike? Go watch that last episode. Is if you know, you know. But the uterus worked so hard, to get the uterus get itself ready for implantation and pregnancy. And then we talked about, but then a lot of the times it doesn't happen. What happens as a result? But this episode, we are going to talk about if fertilization does happen. We're going to be talking about okay, so fertilization does happen. What happens in that little zygote? And we're going to talk about all the things that the MCAT's going to test you on and expect you to know and how they're going to test you on it. So we're all going to be talking about all about fertilization all the way to birth and how the MCAT's going to test you. I'm super excited about Molly. I don't know you. I love these build-off episodes because we talk about one and we're like, okay, let's continue the story on.
Yes, so much fun. And especially this one, it builds so much off of our menstrual cycle episode from last episode, of course, but also we're going to see some callbacks to our meiosis and gametogenesis episode and even back to our endocrine episode, which I don't even know how long it was.
Months.
So it'll be super fun. And ultimately, one thing I want to stress before we really dive in with this process is how the MCAT likes to test it. Okay, because when you Google, what happens from fertilization till birth, you're going to see a lot, right? And when you're in med school, you're going to learn a lot about organogenesis and all of these extra stages that happen during the different trimesters of pregnancy, etc. I want to be super clear just before we hop in of what the MCAT really focuses on when it tests on, embryogenesis and early fetal development, right? It's mostly very early, first 3 weeks, when this is still really a microbio situation, right? As we start to really grow and develop and we get macroscopic, right? We've got a full-on baby in there that's, growing in size rapidly. The number of questions really taps out, right? So
that's not a lot.
Yeah. So most of the questions you're going to see are going to start with, again, stuff we've covered. So hormonal cycles, embryogenesis, gametogenesis, meiosis, things like that, up to fertilization. Then we're going to have cleavage and blastulation.
Love that term.
Which blastula formation, if you didn't get exposed to that beautiful term in your undergrad, blastulation. We'll talk a little bit about implantation and then gastrulation, right? Where we form germ layers. Those germ layers ultimately go on and create all of the different types of tissues that we will see in the growing embryo and later baby. And then pretty much MCAT taps out. Okay, you might see a question every once in a while about neurological development, right, with neurulation, but pretty light. We don't really get much about organogenesis. We don't really get much about the later stages of pregnancy. So just something to kind of keep in mind. We really focus from fertilization to gastrulation, and then we really ease up, okay, in terms of the numbers of questions that you're going to get with it. So we'll go through each step one by one. And talk about what is most important. But I feel like it's nice to have a disclaimer because there's a lot that you could dive in with base first if you wanted to.
Yeah, I know. I took an embryo development class when I was an undergrad. So if you took an embryonic development class, there's so much more like about like okay now concentration gradients to like deciding
Yes.
okay what the organs are going to develop. There's like sonic hedgehog, like those
Yep.
sorts of proteins. Like if you those maybe are so they're, like buzzwords that you're like, oh my goodness, I remember these things. MCAT doesn't test those things. It
Yes.
does not. So we're not going to be, this is not a episode where we give you everything that actually happens. No, we are really going to focus on what the MCAT is really going to hit on. So that absolutely, I love that you like preface that because this is one of those topics like, yeah, there is so much depth that the MCAT doesn't really dive in on.
I
So
thought of the same thing in my
yeah.
embryogenesis class. I'm like, oh God, or animal embryonic development, I think is what it
Yeah.
was. So it was
Yeah.
like, I even had multiple different species we were learning about, and I was like,
Mhm.
I need a
Oh, like
quick.
like flies versus frogs and the different segments. I'm like,
Yeah.
none of that. No, if you're listening to this and you're like, wait a minute, what? Don't worry
Yeah.
about that. That's not on the MCAT. We're going to focus on what is. And the first kind of thing we want to focus on is, I think, really, really great, especially because it's kind of a callback to where we left off in the menstrual cycle, which is fertilization, right? Again, big callback to last episode. We talked about what happens when fertilization does not occur. You know, the progesterone worked really hard to build up the that uterine lining, maintaining that lining, all that vascularity, all of those glands ready to go. But if fertilization and implantation doesn't happen, all gets flushed out in menses. However, right, if fertilization does occur, well, what is fertilization, right? This is when the sperm, that gamete from the male, actually meets the secondary oocyte from the female, not the ovum, not the ovum. It's a secondary oocyte when they meet. Because this is what happens when this occurs. This is when that secondary oocyte finishes meiosis too, right? Because this is the thing where, again, this is a callback from not just last episode, 2 episodes ago when we talked, like we were 3 episodes actually, because we did like the holiday episode, where we
Last
talked
year, go back to
last
last year.
year, call back to last year. It's been a whole year when we talked about this, where in male gametogenesis, very, very straightforward. There's no pausing, they just go. But in female gametogenesis, one of the second pause is after release from the ovaries into that space and then comes into the fallopian tubes, gets brushed in there, but it doesn't actually finish until they meet. So this is the first step where once that sperm meets the secondary oocyte is now going to finish meiosis two and become an ovum. Right. So this is now, once it actually does this, where does this actually occur? A big misconception is that this meeting occurs in the uterus itself. Well, is that true, Molly?
No.
No, absolutely not, right? It's
Yeah.
actually, this occurs in the fallopian tube, in a region of the fallopian tube called the ampulla, right? And so I don't know, do we want to like go into like the specific parts of that secondary oocyte, like the zona pallida and the corona radiata? Do we want to go through that right now?
I mean, I feel like we don't need to get in too much in the weeds. I feel like
Uh-huh.
it's suffice to say that around that secondary oocyte, there are multiple layers of
Hmm.
cells that
Hmm.
ultimately that sperm has to travel through and kind of, it's almost like an obstacle course, making its way through, and it's going to reach this layer that's really tough to penetrate. But the sperm actually has a very special region or a special little pocket in the head of the sperm that has acrosomal enzymes that it can release and digest so that it can reach the plasma membrane of the secondary oocyte, which you're right, is not an ovum, not yet, still getting ready, so to speak. I feel like that's probably as in depth as we really need to
Yeah,
go with
yeah.
the corona radiata, the zona pellucida, etc. Know that the sperm requires enzymes to really get close enough that it could fuse with that plasma membrane of the secondary oocyte and ultimately release its nucleus. Not
Hmm.
just nucleus, everyone just says nucleus, but also its centriole so
Yeah,
that the
the
zygote has
zygote.
both.
Right,
Right.
yeah, so because it only has one. So, because that's an important part, like that kind of obstacle course, and it's kind of like it gets to the end of the obstacle course and then just breaks down the wall. It's like gets to the
Yeah.
obstacle course and it's like, oh, let me just let's just break down the final wall. And it's really important. They do act like testing. I think when they do test this topic, knowing that there's this acrosomal reaction, like there's a like end tip of the sperm has this acrosome sac that gets released, because this is an important reason why multiple sperm usually don't fertilize the same egg. Right? Because if you actually look at a picture of, all those sperm for going to the egg, actually multiple sperm meet that obstacle course at the same time. There's multiple in there, but we know it's only one sperm per egg in most
Most cases, yeah.
most cases, right? Because once one finally gets to the obstacle course and releases those acrosomal enzymes to break it down and fuse, what that causes is this reaction. It's like basically the reaction of all of the obstacle course where it's like, okay, now nothing's getting through. Like everything hardens is like nothing else can go through. We had a winner, like there's a race and there's a winner to the race. The one that first releases acrosomal sac. I don't even think it's called an acrosome, but it releases acrosomal enzymes. And then everything else that didn't have a chance to do that, even if it's still mid obstacle course, is not getting to that egg. They lost the race. Sorry, buddy. Second place is the same as last place. And so now I kind of just like to kind of preface that because that's an important part about why it's only one sperm per egg because everything else just gets kind of nothing go. And so once they fuse, right, again, it donates half its nucleus, it donates the kind of that haploid nucleus as well as one centriole to the kind of ovum, which is now it's now an ovum nucleus and one centriole. Now those nuclei form, they go kind of fuse together, and now you have a diploid cell. Right? Because you had two haploids, two half nuclei, I guess, suppose. Then they form into a diploid cell. This diploid cell is what we call the zygote, right? It is now a zygote. It's not a zygote, right, before that point, right? They're just two haploid gametes. Once they form, now we form that diploid zygote. And it's here, right, that like now you can have the whole genome of the individual, right? And so this is the point where you have one cell that's 2N, no more haploid talk anymore.
Yes. And I feel like it's cool to mention why it's so important that we have what we call the cortical reaction, right? Where we release the cortical granules. I'm speaking as the secondary oocyte, right? We release cortical granules to essentially harden that layer around the plasma membrane so that no other sperm could fuse and enter, right? Because ultimately, if you have multiple sperm release their nucleus, you're going to end up not haploid, or not diploid, I should say, because you have two haploids coming together. You would end up triploid,
Or
right?
quadruploid.
That does not make sense,
Or
right? We don't
tetraploid,
want
yeah.
that. We call it polyspermy. In an evolutionary biology sense, it's super interesting. And we actually have very different evolutionary mechanisms to prevent polyspermy. You might learn about them if you take a full course on embryonic development, etc. But many fish actually have fertilization externally. So how do you time that, right? You could have a ton of different sperm approaching a given egg that's free floating in the water. How do you prevent that? They actually have a fast and slow block. Google it. It's pretty cool. Humans, we don't have to worry about that as much because it's a very long path to get there. Again, it's like a marathon and then an obstacle course.
Yeah, yeah. It's
Really,
like a marathon, but the marathon itself is also very like dangerous and stuff. There's also a lot of like things that has to get through, even to get to that point.
I
But
know.
yeah. But yeah,
Very
the MCAT
risky.
doesn't really focus on before like that, like the uterine tract and all those things. It really kind of focuses on this kind of endpoint after this whole journey they've gotten, the final one gets it, and then everybody else, it hardens, nobody else. And of course, it's not a JW MCAT podcast without Molly bringing up evolutionary biology. You
I
just can't
know.
do that.
You know what? It's just cool, okay? It's
You know
cool.
what? You're not wrong. It is cool. But now you form this diploid zygote, right? It's only 2N diploid, right? We don't want polyspermy, at least in this case. And now I think it's important to talk now about to recognize that this single cell is now going to form every single cell in the human body, but not only every single cell in the human body, but also the placenta that will also be present that will also be growing in that uterus. So because this cell, like this one zygote, will form both all of the cells in the human body and the placental cells, which are extra, like outside of the human body. I mean, none of my cells came from the placenta, right?
Yeah.
This is why we call this zygote a totipotent cell. A totipotent cell is kind of like it is the ultimate stem cell, as in it can not only become every cell in the body, but also some of the cells outside. When it can do this, it can do the entire embryo in both. It doesn't stay this way for that long ultimately, but when we say the term totipotent, that means all of the cells in the body as well as the placenta. So this is important to know what we kind of term, what kind of a stem cell this zygote is.
Yes. And we'll track this throughout our discussion today, right? Totipotent, you can create everything,
Mhm,
everything, right?
mhm,
You almost add together or create a total of the body and the placenta. Okay, so if you see either we're talking about that very initial zygote right after fertilization, or if they mention that this cell can become as well as the human body tissue and cells, but the placenta, you're talking about a totipotent stem cell. Do we really ever see totipotent cells any other time?
No, not really. Like really, no. Like once we start specializing into the body, now it's like okay, extra embryonic tissues like the placenta are just not part of the conversation anymore. So it's really
Yeah.
only at this point that you have the totipotent cells.
Yeah, and
Yeah.
actually, I took a stem cell bio class as well. There's so much that you could talk about there, right?
Yeah.
When you take away the placenta, and we'll talk about this in a moment, or the ability to form the placenta, they're going to be called pluripotent stem cells. And there are
Hmm.
actually ways nowadays with biological and biochemical research that you can induce pluripotency. You could take a skin cell or another type of cell and actually make it into a stem cell that
Hmm.
can create any other type of tissue, right?
Hmm.
There's really no desire to induce that to become totipotent because you don't really like if you were going to try and give someone stem cell therapy, you're probably not going to try and make a placenta for them. like
Yeah,
you're
like
trying
we
to
don't
take
want a new placenta. Like you don't have.
yeah, like if you're born, you're probably fine, right? Like you might need stem cells to treat a muscle tear or something, right? Like
Yeah.
who knows, whatever you might need it for, but like you don't really need
Yeah.
totipotency
Hmm.
except for
Hmm.
this one cell, the zygote, really, and a couple of those early divisions. And I feel
Right.
like here it's also important to mention, call back to gametogenesis, this egg that has now been fertilized, this ovum, and now a zygote, of course, is huge.
Hmm.
It is huge and it's chock full of organelles and RNAs and proteins and all of these things that are going to be required to grow that new embryo, right? From that zygote, that individual cell. And this is really important to mention because when we talk about sort of the second stage after fertilization, what pretty much happens, not literally immediately after, but very, very soon after, we basically take a giant, like, axe and just cut this cell into pieces, right? We call it cleavage. We don't grow really at all. Normally, when we talked about cell cycle, you go through interphase between mitosis, right? Like you pause and you grow and you develop, right? Of course, you're still going through interphase. You still have to go through, for example, the S phase. You need all of your cells to have a nucleus. But in this early cleavage phase, we're not really pausing to grow the cytoplasm and make new membrane and make new organelles and new RNAs and proteins. We do this very quickly, and we're essentially just dividing up the resources that were already present in our extra, extra, extra large egg. It's like taking a giant building and then cutting it up and making it into a bunch of apartments. Like
Ah.
it didn't grow at all. It's just now
Hmm.
being divided into different little areas, right? Different
Hmm.
cells from that. So that's cleavage.
Hmm.
And I feel like it's important to talk about what the or what the egg or the ovum looks like.
Right.
From
It's
here.
big. It's
Yeah.
it's big and then yeah, once it's from that zygote. And yeah, you're totally right. It's like the same size, like it's like a ball, but now you're just cutting up. And these phases of cleavage, these are just rapid mitotic divisions, right? It is mitosing. It is one nucleus becoming two identical daughter nuclei. But everything in the like cytosol, like everything that's extra outside of the nucleus, that's what's being divided, right? Like that's what's kind of being cut up, but no new organelles are forming, right? It's kind of like just kind of this all being cut up, no more space. So there's these rapid mitotic divisions, so it's like cutting in half, and then a number of cells increase exponentially. Right,
Yes.
because first you have like that's like exponential growth of cells. Because first you have one, you cut it in half, now it's two. But then you cut both in half, now it's four. Then you cut all those in half, now it's 16. Right? It's kind of like or eight, then 16. Right?
Yeah.
Like yeah, I'm sorry, I missed a number there. So all of these now you're growing exponentially, like 2, 4, 8, 16, 32, 64. Like you're constantly going up and up and up. And so it's this rapid, like just like we have more and more cells, but because the total cells themselves, like the total amount of cells, it's all the same size, each individual cell gets smaller and smaller and smaller each and every division. And so these cells, right, that are forming this ball of just a bunch of different cells are known as blastomeres, right? These are tiny, tiny cells known as blastomeres, and this solid balls of cells of blastomeres are known as a morula, right? Funny name, I know. That's what our zygote has now formed, is into this ball of cells that's a morula. And remember, this fertilization right? This fertilization of the sperm and the egg happened not in the uterus, but in the fallopian tubes. So although they met, in the fallopian tube, they're now still traveling to the uterus, right? Toward the uterus. All of this division, all of this, fertilization, cleavage, we're not in the uterus yet, and we haven't implanted yet. We're still on the way traveling. And it's for this reason, if we're to talk about like timelines, all this cleavage and early division happens in the first like 5 to 6 minutes, right? As soon as fertilization, this is like very early on, not like immediately. It's not like in the first like 3 seconds, now you got a morula. It's like in the first 5 to 6 minutes, this occurs and it's traveling towards the uterus, and you have this ball of teeny tiny cells that are the same amount. So now, what happened next, right? We've got our cleavage, we got this ball of cells, this morula. Now what happens, Molly?
Yeah, so we're making our way closer and closer to the uterus. We're traveling through the fallopian tubes, and we start immediately, right? But ultimately, it takes some time, right?
Mm-hmm.
What we see is around day 5, as we continue to divide and divide and divide and divide, without a ton of growth, getting these teeny cells, we've got this giant ball, which you said is called a morula, which is a weird name, but kind of cool, right? During everyone's favorite process called blastulation,
Blastulation, absolutely.
which we're making light of this and fun of it because I never heard that term. And yet that is exactly what it's called. And we have gastrulation as well later. So blastulation, it's perfect. Like I
Hmm.
love that this is a term because we're going to form a blastula, which is a ball of cells, but with a fluid cavity in the middle. So it's not quite hollow. It's got regions where you've got a pretty solid mass of cells. We're actually going to call it the inner cell mass. But then we also have this big fluid-filled cavity within that ball of cells. So it's partially hollow.
Yeah.
A weird way to phrase that. It's like imagine a water balloon, but with like sand at the bottom.
Oh, that's a cool. I always like visualize it as like, it's like the eyeball, or it's like the eyeball is like, oh,
Mm,
there's like most of it's fluid, and then at the front you have this like area of like
I
lens.
like that
That's
too.
the inner cell mass. Like
There
that's
you
how I always
go.
visualized it. But yeah, no, it's like they're not. It's not empty space. It's just like fluid in it. Like, but there's no cells there.
Yes.
Yeah.
And
Yeah.
so this is I always thought it like, what's the point of this? It will become clear when we talk about gastrulation, where we're going to start to see a lot of migration of cells, right?
Hmm.
But at this point, during blastulation, we're basically forming this fluid-filled cavity, right? Which will eventually, right, help create not just, again, if we talk about the blastula, not just the inner mass cells that will become the embryo, right, and will
Hmm.
become the new human body, but also things like the placenta, right? So
Hmm.
it's the setting where everything is going on, right? We need that
Yeah.
fluid-filled cavity to help form those tissues later. Um, Again, that's about day 5, and at this
Yeah.
point, it's important to mention we've already sort of specialized. As we've formed this blastula, we're getting very early, we're starting to differentiate between, oh, okay, I'm going to become the placenta and I'm going to become the embryo, which means our potency is changing.
Absolutely. We're not totipotent anymore because now we've already sort of specialized those cells because now we've separated the trophoblast, which is like the outer lining from the inner cell mass, which is this is mass in the middle. Those cells in the inner cell mass can't become placenta anymore because they've specialized. So now these cells are no longer totipotent, but they are now pluripotent. As soon as we have formed those inner cell mass, those are pluripotent, and now because they can't become the placenta, right? And so it comes from those trophoblast cells. This is kind of where the MCAT kind of like forgets about the trophoblast. Like they know, like, okay, the trophoblast is there, but like the MCAT doesn't really tell us, like, oh, what's the trophoblast going to do? It's like, okay, that becomes everything that's not the embryo, including the placenta as well as other structures. But the kind of real star of the show here, where the MCAT really likes to test it, is the inner cell mass, because that's where, like, again, the human body, everything in the human body will come from that inner cell mass. But because it's kind of specialized, it is now pluripotent, not totipotent.
Yes. And that's exactly why, like, again, not really MCAT relevant, but for stem cell therapy, when they're trying to create what we call iPSCs, induced pluripotent stem cells, you don't, pluripotency is amazing. I could make that into a myocyte. I could make that into a neuron, hypothetically. I could make that into a skin cell. Like I can make whatever I want, right? These pluripotent cells can make any cell of the human body, right? Because they are so generalized, right?
Right?
Um, but not as important to go all the way to totipotent.
Right.
So at this point, we've got some very early specialization. We're starting to become unique and starting to get these sort of specialized functions, right? And we're making our way towards gastrulation, but first we got to check in with where we are. Okay, so at this point, we're still in the fallopian tube and we're getting close to the uterus. Okay, at a given point, and this is typically about 6 or 7 days after the fertilization event, you're actually going to have the blastocyst, right, enter, or the blastula. You're going to enter the uterus and then embed into the uterine wall. Now, keep in mind from our last episode, when we were talking about the uterine cycle, we've got a thick vascular lining in our endometrium. Okay, so this blastula gets to just kind of burrow in there, right? And now have access to so many different nutrients, resources, all of this good stuff. Okay, so it's implanting or embedding itself into the uterine wall. And at this point, we've got some pretty cool hormonal stuff going on.
Yeah, absolutely. This is like the major callback to like last episode because remember we were talking about last episode like, oh no, implantation did not occur. All right, let's just shed everything. Let's just throw it all. I worked so hard, but
Tear the walls down.
tear the wall down. But hey, look, implantation occurred. So wait a minute. If I have this blastocell or blastula that implants in my endometrial lining, do I want to be tearing down the walls now and getting rid of this cell?
No, that would tear the zygote away as well. Or it shouldn't
Yeah,
be zygote anymore. It's a blastula, but still.
it's our little cute little embryo, right?
Yeah.
So that's why we need to make sure that our uterus doesn't flush this away. We need to keep this blastula and the lining still there. So upon like implantation, this is where we form like the trophoblast becomes, or the trophoblast releases HCG. Now, this hormone is really important. We didn't talk about it last episode too much, but this HCG will target the corpus luteum. Recall, the corpus luteum has a little bit of, as soon as ovulation occurs, the corpus luteum is formed from that degraded follicle, or from that burst follicle, but it's on a little bit of a ticking time clock, where it's going to be producing progesterone, but if nothing happens to that corpus luteum, it's just going to degrade and then go away, and progesterone's going to tank, and then shedding will occur. However, the trophoblast will release HCG, this really important hormone, which will target the corpus luteum and essentially rescue it. Once HCG targets the corpus luteum, it's now the corpus luteum is like, oh, I'm not degrading anymore. I am totally fine, actually. No, we're not, no ticking time. I am here. And as long as the corpus luteum is still there, progesterone will still be produced. As long as progesterone is still produced, our wall will be maintained. There will be no drop, there will be no shedding, right? It's going to be maintained, and our blastula will stay on that wall, which is what we want to see. But this is actually what keeps our progesterone from tanking and from menses occurring upon implantation.
Yes. And it's important to mention that HCG is really not produced any other time. Right. This is something that's very specific to the very early stages of pregnancy, right? Like you would not have this if you did not have implantation. And this is actually how pregnancy tests work. Because this is really never something that you would make if you were not pregnant, because it's secreted by a fertilized egg, right? It's secreted by that early embryo after implantation. If you have HCG, you probably are pregnant, right? That's how pregnancy tests work. It's super cool. Again, like you can, maybe you've interacted with that in the past at some level, but like this is how people, whether they're scientists or not, can determine whether or not they are pregnant. Like it's so fascinating. I love talking about when like this crosses over from something that just feels MCAT related exclusively to like, wait a minute, a lot of people engage with these types of products or use something like this in their life. And it's literally, in this case, just detecting HCG. That's it.
Right.
So super, super interesting. And at this point we have implanted, okay, we're saying to the body, hey, hold on, do not shed that endometrial lining. Okay. We need that. We don't want to tear down the walls. And at this point, our embryo is ready to take the next step. Right, which is gastrulation. And really, for the context of the MCAT, this is a pretty big step and also one of the last that we're really going to care about. Okay, not that we don't care about it, but as far as studying for the MCAT, this is This is the last big thing that's pretty testable. And this is essentially the idea where that inner cell mass, which we'd mentioned earlier, that's part of the blastula, is going to go through some interesting migratory steps and ultimately form what will become three germ layers: the endoderm, the mesoderm, and the ectoderm. Now, it's pretty common to get questions about what types of cells derive from each germ layer, but it's interesting, and I feel like it's fun to look at a diagram of this with the migration. Hard to describe, but basically these cells are going to migrate and they are forming these three layers, which then go on and create all the different tissues of the human body.
Right, right. And it's so cool, like when you map it out, like based on that migration, what ends up happening the whole body. Because how I usually think about like the three germ layers, like we talked about ectoderm, mesoderm, endoderm, is ectoderm anything that's on the outside. So I'm thinking like skin. I'm thinking like, yeah, just mostly the skin, right? And some sweat glands here and there, like the upper layers of the skin. That's the ectoderm is outside. Endoderm is any lining on the inside. So that's like my GI lining. That's like my lungs. Like those are the two like inside linings that are really important for the endoderm. And then I think mesoderm, everything in between those. Like it's kind of like between the skin and that inner lining, everything else is essentially mesoderm. There are kind of two, like there one is like really important, like a really good distinction though, and that's the nervous system. Right. When we talk about like, so ectoderm is like skin, endoderm, inner lining, mesoderm, everything in between. Nervous system, you would think just based on that is like, oh, mesoderm. It's actually not. Ectoderm actually derives, or so nervous system actually derives from the ectoderm, right? Because we have the formation of the neural plate and those neural crest cells, and it's from the ectoderm that the nervous system actually arises. And this, I think this idea alone is like a really testable topic. Like, I think, when we talk about embryo development, I don't think this whole topic in general is like, the highest yield thing on the MCAT. But of everything we talked about, like this is just one of those topics that really consistently get tested. Not on every single exam, obviously, but when this topic is tested, they do like testing, where did the nervous system come from? And that is from the ectoderm. But other than that, like everything else is like very general, like mesoderm, everything between the endoderm and ectoderm, endoderm, inner lining. As long as you have that, that's like 99% of what these questions are going to be about.
Yeah, and there's tons of mnemonics for this. The
Mhm.
one that I hear the most is atractoderm for ectoderm. And
Uh-huh.
it's kind of funny because it's like, okay, well, if you're talking about just pure attraction, why are you attracted to somebody? It's typically skin deep or their brain. Hopefully it's them.
Oh, I have never heard of that in my life.
I thought
That's
that
so
was
fun.
kind of cute. Atractoderm, most of the ones when you Google it, like, or you look on Reddit, like they're like, oh, it's atractoderm plus the brain or plus the nervous system. And I'm like, hey, they're like
Wait a
everything
minute.
that's like just your skin and then plus the brain. I'm like, come on, someone's brain can be attractive. I kind of tweaked it.
Of course, of course. Well, I
Yeah,
love how they're like
like
attractive plus the brain. Like,
don't
no.
take it
That's
too far because you're not going to see the heart as part of the ectoderm, right? They can have a good heart, but that doesn't make it attractoderm, right?
Yeah, no, that's very mesoderm in between. But
That's
yeah,
very
no,
mesoderm.
I have never heard tried. That's hilarious. I
You've
love
never heard
that.
of
I have
tractoderm?
never heard of tractoderm
Oh
before.
my gosh. Yeah, that one I heard so often and I really didn't like it because I'm like, that doesn't even explain the brain. And then I was like, actually, you know what?
You
It
know what?
explains the
I can
brain.
make it. I can make it that way. But yeah,
Yes.
like that, like this ectoderm, mesoderm, endoderm kind of separation. Yes, very important to know these differences. But again, I don't think it's. I think a lot of people just like try to get a flashcard and memorize them. I think this is very like conceptual to understand. I think it is good to kind of take into account. And
Yes.
so. I think when we kind of really talked about like the whole pathway, like I think because now that we've formed these three layers, now this is when like the MCAT doesn't really test too much after this point,
Yeah.
where it's like now, okay, well these are going to develop even further, right? These mesoderm, like the cells in mesoderm, will further specialize into the heart cells versus the muscle cells versus all these other cells, right? There will be further classification, like specialization of these pluripotent stem cells into once they become more specialized multipotent stem cells. But again, the MCAT doesn't really test those parts that much until birth, right? And then you can talk about lactation, or not lactation, right? Like oxytocin and birth and contractions and positive feedback loops. That's something we talked about in the endocrine episode. So you should definitely check that out if you are like, oh my goodness, I want to hear about positive feedback loops. But when we're talking about how this is actually tested, what we've talked about here, a lot of the times they test on like what happens when this goes wrong. So, for example, they say something along the lines of, oh, say we had an issue with neural development. Which of these cell lines probably went wrong, right? And so, then they'll be like, oh, mesoderm, ectoderm, endoderm, and then plus like trophoblast. And then they'll be like, oh, and you'll be like, oh my goodness, I don't know which went wrong. And oh, it's between mesoderm, right? No, this is an ectoderm kind of question. Or they might have questions along the lines of, hey, we have a sperm that can't release its acrosomal enzymes. What's going to happen? Right? These are the types of questions. We're like, oh, no acrosome? Oh, it can't actually penetrate. Therefore, no fertilization can occur. Where the questions that they ask, especially on processes like these, and a lot of the processes that we've talked about in the last few episodes, are not questions that you can straight memorize all the time. These are ones where it's important to understand the process, why each step occurs the way it does. And when you understand it that way, like, okay, the flow, it actually makes a lot of sense why this process is like that. But I think what makes this topic a lot of times one that students I think tend to ignore because when they look it up online, there's like all these details and they're like, oh my goodness, I don't like this. Avoid, avoid, avoid. I'm not doing this. But that's why if a big takeaway from this episode, I would want anyone listening is understand these steps. When is a totipotent? Why does that make sense? It's totipotent. When is the pluripotent? Why does that make sense?
Yes.
Mhm.
I think it's really such a cool pathway. I mean, really, we kind of spent 3 episodes breaking down reproductive system in general, right? With
Yeah, yeah.
hormonal sequences. But I feel like reproduction is something that has so many callbacks to so many
Mhm.
other layers of biology and biochemistry that it's so awesome, right? And you do
Mhm.
get questions, maybe not exclusively about this material, but how this material connects to other topics within MCAT scope. And that's why it's worth it, right? Not
Mhm.
only is it cool and is it important to know for med school and beyond, which by the way, you will learn all of the stages
So
of
much
human
more. So much
development
more.
or embryonic development and pregnancy and all of those things. You'll learn it in med school. Not only is it cool, but it also helps you understand other areas of MCAT content. So kind of fun, right? And again, it connects to cell cycle, mitosis, meiosis, gametogenesis. It connects to hormonal regulations. It connects to stem cells, basically cell biopotency, all of these things. It's kind of a cool process,
It
right?
is really that I think is very relevant for anyone going to med school. This is something I think this is one of those topics where it's like, if you don't like this topic, why are you going to med? And kind of like it's kind of like one of those
Cool.
topics it's so cool. Like anyone who is going wants to go into medicine, this is a topic you will learn so much more. So this is one to understand foundationally now, so that
Yeah.
when you get to that point, you're like, oh, I already studied all this and I understood it. Not
Yes.
oh, I memorized it and got super overwhelmed.
Yeah.
So that's why the topics like these are super fun, just like
Yeah.
all the topics we talk about, and including the topic that we're going to be talking about next episode. Because we've kind of really talked about like, fertilization. We've talked about okay, all the menstrual cycle. Now the cells that will soon one day become that, little baby will form. But one of the most important structures that are in the human body that we kind of refer to a little bit at the end there, or we focused on, is the brain. This noggin right here that's super attractive, right? From the attractor term.
The
Um,
attractive term.
one thing that's really important is that sometimes the brain anatomy can be really overwhelming. You're like, oh my gosh, which part is this brain? What does this part of the brain do? What does that part of the brain do? Yet it's one of those topics that's so testable. And that's why we're going to be talking about it next episode, where we're going to be diving into like now that we've formed, we've decided which cells are going to become, those ectodermal cells will become the brain. Well, what are the structures of the brain? What will actually form? What's the purpose of each structure? Like, how is the MCAT going to test these? Because ultimately, the brain is something that's so complex we don't we like the scientific community doesn't even understand it. Well, then how are we for the MCAT supposed to do it? Well, we're going to be talking about that next episode. I'm really excited for it.
Yeah, that'll be a fun one. It'll build off our original episode of the nervous system, but this gets fun because there's so much overlap.
Hmm. Hmm.
We'll have fun because so many students hate learning about this, given that a lot of these regions of the brain have similar functions or related functions, or maybe one function actually involves multiple parts of the brain, especially when you talk about movement and sensation and all of these things. So super fun. Very excited for our next episode. We hope you enjoyed this one. And we can't wait to catch you in the next one. Until then, happy
Happy
studying.
studying. Oh, that was almost, it's like